So we'll move on to the next speaker. He's Dr. Narinder Dhillon from AVRDC. He's doing breeding work there. Again, he's going to talk about the bitter gourd varietal selection and multi-locational trials. Dr. Dhillon?
Thank you very much. Good morning to everybody. So this work which I'm going to present today is from our this BMZ project allotted to AVRDC 3 years ago. We are about in this 12 collaborators in this study which I am going to present, and I am simply the presenter. These are the workers here. So this is the project which was allotted to AVRDC, under which we performed this work, and we had a lot of collaboration from the seed companies for this work. So there are many questions from the bitter gourd to the breeders and the nutritionists. There are many myths, and some of the questions and the myths we try to answer through this project. For example, AVRDC recently released high-yielding bitter gourd lines which are yielding nearly to the hybrids, but they're low in bitterness. And in Bangladesh, one of the person in the audience said, why you breed low bitter? More the bitter, more antidiabetic compounds. Where is the scientific evidence? Nowhere. Why you breed light color? The more the dark green color, more the antidiabetic compounds. Where is the scientific evidence? No. Why? So there's so many questions. Are white color more in antidiabetic compounds? Are dark green colors or light green? Are the varieties more rich in antidiabetic compounds? Or are the smooth one more? So all these questions will be answered in this forum. So we have one output too. In this we try to see the germplasm collections in the AVRDC. What's their horticulture worth and what is their nutrition worth? If it is some promising, can the breeders use in their breeding programs? So one of the objectives was this. So we choose from these countries 53 accessions, and mostly from Bangladesh of India, because it's the primary center of diversity. So we made this set of collections like this. Why? Because we want to answer the questions. What is the association of the color with the antidiabetic compounds? What is the association of the skin pattern with the antidiabetic compounds? What is the association of the fruit shape with the antidiabetic compounds? So that's why we made a germplasm assembly like this. So for horticulture evaluation, we use these traits for the flower emphasis, for the maturity, fruit yield contributing factors, fruit numbers, average fruit weight, yield, etc., and vine vigor. There are many questions breeders also. Is if there more vine vigor, is there more yield or not? So we will try to answer here these questions. Then various fruit traits we choose. So this was the classification we do for the fruit skin colors, 5 classifications. Then we have classification for the fruit surface. Then vine vigor assessment. Less vigor, medium vigor, high vigor. This is the judgment by eye judgment by the breeder, because he used to screen large populations for that. Then this was at the Shanwa, the weather data in which we conducted this study. And for 2 years, we found that there was a significant entry into year interactions, means the entries didn't perform same manner in the years, and also the ranking was different in the seasons. And the year has also the effect. Now the performance in the 2 years, and I want to emphasize here is that this experiment was in cages and the vines were pruned and the yields were less. Had it been like Indian conditions, the yields will be more. So I just present the yield data. So there are the lines which are quite high yielding from the gene banks. And from these 53 lines, on the basis of the nutrition data, we singled out 25 lines for the next year. And this is the pooled mean. This is the difference. The less the difference, more stable is the line. That's the difference. More stable is the line over the years. This 1850, 4785. So this is the horticulture data. Now you remember this data, but when you present her data, try to correlate the antidaptic and nutrition qualities with this data. Now this is, you can judge the stability. TOT 850 very stable, and 4785 also very stable. Now how they look like, because the breeders also when want to order some lines from the gene banks, they just also want to look the shape, color, what they are going to order, because there is a segment-wise breeding. So how they look like is this, and we are using these lines for our recurrent selection program, using elite lines as well as gene bank lines. This is our second phase of breeding at AVRDC. Now there was another trial, there's a multi-location trial, in which we used the 20 hybrids of 11 seed company. These are the market leaders in India and other countries. So why we want this study was, at the multi-location, we want to see the performance of these hybrids. If in our nutrition study, some hybrids found very good for the antidaptic compounds and other nutrition qualities, then we can associate that at various locations, how is their yield performance and other performance. And can we use those hybrids for your breeding programs for subsequent derivation of lines which are rich in antidaptic compounds or not? So this is again, when we made this germplasm assembly, we kept in mind the variability of the fruit rates, because again we want to see the association of the fruit characters with the antidaptic compounds. So we did the horticulture evaluation as in the output 2, same. And here is the combined ANOVA at Shanua place. And you see there's a significant difference between the hybrids for all these horticulture characters. Yeah, at Ludhiana. And then we have in the Shanua, this place, you can see the entries also have a significant differences, but year doesn't have here the difference and no year into interactions. And this is at the Hyderabad location. You can see again the entries have significant differences for the various fruit characters, and in some for the fruit weight and the average fruit weight, and this fruit width there was the interaction but not for others. And now this output 3, let's compare for 2 years at Shanua. This is a cage study, not in the open, and the vines have been pruned. Now here you can see Abhishek has a very high variation over the years, less stable. Aman Shiri is very stable. VGCT 385 very stable, unstable, very stable here. And come over to Hyderabad, you can see here the large number of differences over the 2 years. Because in the first year, the crop was not very good. There were so many constraints of the environment, of the diseases, and by the second year, the crop was much better. And this is how it performed in Ludhiana, all these hybrids. ARBHT1, very stable in this performance for the yield, and also VNA 28, very stable for the yield for the 2 years in the Ludhiana. Now this is in Arusha. This is a very good study, and the average yields were very high, 34 tons per hectare. That also gives us idea that this crop can be grown in Africa very well. Start with Tanzania, very good. So bitter gourd can grow very well there, can yield very well, can get the optimum potential of the lines you can get over there. Now, what are the ranking of the hybrids? Because many people are from the industry who have contributed the seed of these hybrids. They are very much interested to know the performance of their hybrids there. So what is the ranking of the hybrid over those locations? Now if you say general here, the first out of these 20 hybrids, what are the first among 6? Pali is coming here in Arusha among the 3, then also Pali in Hyderabad, also Pali in Ludhiana, Jasper at 2 locations, Amanshiri at 2 locations, Ben 455 at 2 locations, likewise. But this is not a sensible comparison. Why? Because comparison should be segment-wise. There are different segments for the different markets. They are not comparable. So we try to do that. How? Now this is high-yielding hybrids at the 4 locations. You can see their pictures, but now we try to segment it. Now this is the green, dark green, and this is a medium South Asia type. This is small, VN28 New Moon. Then this is a creamy white, these 5 hybrids. Then Southeast Asia type. These are 4. Now make a comparison of the yield among them, within them. How is that? Now this is for South Asia type. Green, dark green, warty, and medium fruit size. And here, among the first 4, you can see the polys at the 3 places, Zina also at 2 places, NS 10 to 4 at 2 places, Amanshiri at the 3 places. So this is a clear picture because it's a segment-wise comparison. Then see their stability over the locations. Poly has, I told you that Shenhua the yields are less because the vines are pruned and they're grown in the cages. Now here the poly, very good stable, and also you can see NS451, then Zina at 3 location, very good, then Abhishek also. So likewise, we made for Southeast Asia type this for the light green color, ribbed, and then this long types. So now here you can see that VGC 358, 2 places, Jaspers at 3 places, Ben 45. This is the ranking. And this is the yield stability. VGCT 358 is a highly stable. Then South Asia types, which are cream in color. Now this is US 33, very good at 4 locations. Then IT 465 at 3 locations, Indo-American hybrid. And then High Moon there, ARBHT from Anchor is there. Then this is their stability, this is a very high stable US 33. Very less yield at the difference at the 3 locations. Shenhua yields are always low, I told you, because in cages. Then this is a small type. Only 2 hybrids were there, Vienna 28 and New Moon. Vienna 28, consistently good at all the 4 locations. See, graph, Vienna 28, New Moon, come down. So these are the average yields at the locations of all the hybrids. So we can go very nicely the bitter gourd in Tanzania is a good point here, very good high yield. So bitter taste, I want to tell something. This is about the compounds, and we try to assess the bitterness at the various locations. Now you look at that. We made 3 categories, high, medium, and low. And we trained the persons to do like that. But you can say that in Ludhiana, there is a no low, medium, low category. Only 2 categories they can evolve. But in Shenhua, they can see all the 3 categories and uniform in 2001, 2012. In Hyderabad, they have all the 3 categories. But the categories change over the years. And Arusha will have 1 year data, but all the 3 categories. If we remove this Ludhiana data, I want to say, can we tell from the hybrid, this is high bitter, this is medium bitter, this is low bitter. Now tell us the data of the nutrition. What are the antidiabetic compounds? That was my purpose of doing this classification. So now you see, remove the PAU data, see the consensus emerging. Poly is medium bitter. BGC 725 high bitter. US 33 medium bitter. High Moon low in bitterness. Now compare this data with the nutrition data because there's a consensus here. Now how their fruit look like? Because the people ask me, dark green are the high bitter, not white. Sorry, you are wrong, they tell me. Now what's the data says here? Look at that, high bitter, cream color, highest bitter in these collections. Low bitter, light green, medium bitter, dark green, and also the cream color. Then, We have another study in which we try to see the skin color pattern and bitterness. Now you can see the high bitterness is dark green, green, warty. Low bitterness, they are also green, light green, but mostly ribbed. Another study, high bitter. You can see all the categories here. Low bitter, all the categories here. But high bitter, all warty, no ribbed. So another study, medium bitter, dark green, light green, all, mostly warty, but one ribbed. So what is the conclusion from this study? The conclusion is there appears to be no association between the fruit bitterness and fruit colors and skin patterns. However, accessions with ribbed fruits were either low or medium, but not high in fruit bitterness, and high bitter accessions were always warty. Now, vigor assessment we do with the visual observations, and then we try to see whether the vigor is associated with the yield or not. Breeder says, but there is no scientific evidence. So what we do is, you see, in Shanwa, in the cages, we have exactly the same classification over the years. In Hyderabad, 3 replications. In 2 replications, same, sometimes one change over the year. In Ludhiana, change over the year. So we try to clean this data. How? I say if it's LMM, let it put M, avoid L. Because in 2 replications it is MM, in 1 it is low vigor. So like that we try to clean the data. But this is the most authentic data. Replication-wise, no difference. Year-wise, no difference. Then let's see how we come out. Then we refine the data. Now it comes like that. But still the variation over the years. Here, no variation. Then we see that, say, you can see here, the eye judgment is wrong in one classification. There is no judgment like high or low. 1 year high vigor, other low vigor. There is only one classification difference, medium or low. So then we said that they have an error or they couldn't judge between the medium and the high. Then for the correlation we try to remove this class. Let's remove the class of medium. Remove this class. Then take the Shanwa data, which is same replication-wise, same year-wise. Then what is the vine vigor was correlated with yield. Absolutely true. So what's the conclusion? In the gene bank, we have the accessions which are high yielding, whether they are rich in anti-adaptive compounds, Ray you will tell you. Then you can decide whether you can use in the recreate breeding program or not. And then the difference between the commercial hybrids over the locations for the various horticulture traits, but what is the difference for anti-adaptive compounds, Ray you will tell you. And then there is no association between the bitterness and the fruit color or skin pattern. And the vine vigor is positively associated with yield. Breeders can with confidence use this trait for their selection. We acknowledge the German government's funding for this work and all these seed companies who generously supplied us the seed whenever we required. Thank you very much.
Is there environmental role for degree of bitterness, like if there is strong hot summer, so the intensity of bitterness is increases, and if cloudy weather is there, degree of bitterness will reduce. Apart from the fruit color or what type of what color, is there any?
I cannot say with this because I don't have a data. In Shanwa, for the 2-year data, we have almost the same temperature except the rainfall, almost the same relative humidity. And over other 2 locations, I told you, there is a variation between the category which the person who classified that. But I have not yet the data of the climate to associate with that variation with them. And in PAU Ludhiana data, we don't have a classification of a low bitterness, so I don't consider that authentic data for bitterness. So, but I will come to your question later when I get the environment data of Hyderabad and the environment data of 2 years of Arusha, then I compare the data, then I will revert to you. Okay?
If it is directly correlated with the bitterness,
Please sit down and use the microscope.
if we correlate the bitterness with the colors, so exactly what type of color will responsible for the bitterness?
There is no association between the color and the bitterness.
Actually, if you look, you have already cultivated the protected cultivation and open field condition. There is not at all any the bitter differences is there. So environment not much effect, though little bit effect on temperature is there, but not much effect, significant effect is there.
There's a question here.
Yeah. See, when you are trying to correlate these data of locational differences, I feel strongly that when you are talking about the bitterness, you have to attribute the factor. You cannot generally classify low, medium, high bitterness, and same variety will be differ in bitterness certainly. I agree with the person who raised this question, because most of the secondary plant metabolites will express it in a different way, because even when the slight changes in the temperature or the other factors also. So the bitterness principles, basically the polyphenolic compounds, will be metabolized actually based upon the environment also. So there is a locational differences also. Even within the country, we could see some of them. See, this country, for example, has been mapped as 21 agroclimatic zones. There will be a differences. So probably you should see that you must classify the bitterness for that location also. Then probably you should correlate. Then only you will get an answer.
Yeah, good. The association which I said with the color with the bitterness is only pertains to the one location, Shanwa, where I trained my research assistant how I trained and how I make sure that he's right. How? I give him 3 different fruits of different varieties, which I know they are high, bitter and low. And then I ask them, you taste and tell me which is a high, which is a low. Absolutely, he was correct. So I standardized that. And then the same person I used for 2 years. Of course, there was no difference in the climate or environment or the humidity or the temperature at that place. Only using that location data and that conditions, that correlation which you saw like that is that. Now your second is, for example, at Hyderabad. And I don't have a data of Arusha yet. I can only talk about Hyderabad. And Hyderabad, there is a difference. The varieties, they sometimes, in 1 year they say it's a medium, another they say low. But they never say that this is a high and this is a low. Never there is a change from high to low. Not a single instance in any location. They say in 1 year it's a high, other low. They say it's a medium and low. They say it's a difference of medium and high. But they never say it's a high or low.
There's a question here. Yes.
I'm just wondering, Dr. Dhillon, if you ever have considered looking into the dry matter content associated to
Dry matter content data is available with our nutritional lab. They have done it. They will present it.
Yeah. How you measured this bitterness, means whether you quantified it or it is just a organoleptic test?
Yeah, good. So how I train my person is like that. Even my RA in Taiwan also, for other studies I showed you that. So we chop the marketable maturity fruit, and then all the lines had to be tested at the same time. Not one today, few tomorrow, few. No, same day by the same person. And in between, he has to wash his mouth with the water. It's a very thankless job. And my research assistant very unhappy with me. And I had to give a box of chocolates after that to her.
Means if we get a quantified data, that will be more reliable, because
Well,
it means human errors are.
you are right, but I didn't do that. It's by taste. But for example, what about all your consumers? You go to Punjab, you go to Haryana, they will always say, this is a bitter, this is a less bitter. At least they can make some classifications. Consumer, and we are targeting consumer.
How you just observe the wine vigor? Because wine vigor is the varietal character. Generally, wine vigor, when we consider the last 5 internodal length
just consider and based on that you draw the conclusion which one is the more vigorous and which one is the less vigorous.
Yeah. For example, up to 8 nodes, this much size, you just see the girth, diameter of the stem and made impression in your mind. And when you have for example, to my people, I give the 3 hybrids. I say, you grow it time and again, you say it's a very high yielding, but it's a very high girth. See the medium, see the low. But at some points, in some situations, the people were confused in locations. They were confused between the medium and the high. So to make that correlations, I said they are confused, but they were not confused at one location, repeatedly over the years and repeatedly over the replications. I used that data. But for others, and for that data even, I removed the medium class. Now they can always say this is a low vigor, this is a high vigor, this much thin, this much strong stem. So then the correlation using this high vigor and low vigor and the yield, this correlation was detected.
I'm not talking about the just early stage of the internodal length or the just medium girth. I am talking about the last internodal length.
No. We measure the vine vigor only from the lower, 8 node stem. Why? And we measure when. When the productivity is full, plant has developed branches, everything. That time, the lower 8 nodes girth, how much thick?
There are 2 questions. I will go ahead. Jen can talk to him in AVRDC, but you can ask a question. Go ahead.
Thank you. Regarding the bitter taste evaluation, I just looked at the data on the PAU groups that have the high and median, and then the Taiwan group and also the South India Hyderabad group have low, and sometimes the taste will relate to the sensitivity of our tongues, the individual persons to the bitter taste. So maybe this is also a problem. Maybe we need, because the north, I noticed that the diet is less pungent. So the people may be more sensitive to the bitter taste compared to the south, less sensitive to the bitter taste. And maybe we can have a validation test. Maybe one person, for example, like you, have the samples.
So actually the problem arises due to 2 reasons. There are 2 places. They, at Hyderabad, they involved 3 persons. And 1 person they changed the next year. And they didn't standardize giving the 3 examples which they already found it. That this is the medium, this is high, this is low. Now you all tell whether you arrive at the consensus, and edging arrive at the consensus, then I apply them for this test. And in case of PAU, 1 year there was a 1 person, in the second there was another person.
What was the standard in the organoleptic evaluation for this?
I don't know.
So there is a question from the third. Dr. Young took away the question from me, because to talk about bitterness is, in a scientific way, really a marginal discussion, because we know that the bitter receptors in the palatum and in the tongue are totally different, ethnically different. This is an epigenetic phenomenon which can be educated. So I think to embark too much on bitterness is a difficult business, at least when you talk about breeding. Because we know that we need the bitterness for the diabetic people.
No, there's a, no.
What we are doing in the Western world,
Yeah.
we put out from every food the bitterness because people don't want to eat it. But for the physiological process, to activate the insulin process in the pancreatic cells, we need the bitter ingredients.
Mm.
That's the point. So bitterness is a fine philosophical pronunciation of that what we mean, but we need to have a scientific standard. But we don't have scientific.
We don't have. Then how we do it, try to make it a little bit scientific like that. For example, I belong to this South Asia path, and then my research assistant, he is very sensitive to the bitter fruits. He belongs to Taiwan, because their people always like low bitter. And when I taste their varieties, they are low bitter.
Yeah, so sorry, that's an epigenetic phenomenon, how to raise the children.
Yeah.
If you raise them in a sweeter environment, then you don't taste bitter. That's
Yeah.
dependent on the receptors.
Okay, I think there is a good discussion. I think it's good the speaker finished his talk earlier and he got good debatable question. I think, again, I'll just throw my opinion. I think there are 2 sides. There's a problem right now. In both nutrition side, where we don't know officially or scientifically which compound is really triggering the bitterness. There are certain evidences, but whether those are the specific compounds really triggers the bitterness. And also the breeder also using the human, 1 or 2 humans as testing purpose to breed a variety. That's also on the other side. So I think this whole conference on linking agriculture and nutrition will be a good fit, wherein breeders should be communicating with the nutritionist and medical scientists to link that. I think it's happening in your list. You showed that, but I think still there's lack of communication, in my opinion. Again, this is based on opinion, what I see in your presentation and what I see in a lot of discussion. So it is very important in future. We have to, from the nutrition side and the human side, we have to establish the criteria for bitterness. And from the breeder's side, we should use that criteria. But of course, as a breeder, you cannot use that whole scientific model for breeding every variety because it's not practical in the field conditions. So we have to come up with that plan. And I think in Texas A&M, we have developed that, for example, mildness in onions. We developed that, breeders can use that part all the time. The onion industry got $1 billion in Texas because we developed the sweet onions. Of course, in India, nobody wants sweet onions. That's a whole different story. But because people have consumed more onions in the US, we developed that model. So the breeders are evaluating based on a chemical test. And fortunately, in onions, we knew that compound is there, necessary for that. But in bitter gourd, it is a complex phenomenon. So over the years, I think communicating with your nutrition team there in AVRDC and I think others, and with even Germany, I think this problem will be solved, I think. So with that said, again, we appreciate your great lecture and thank you very much for that.
Thank you.